Hello students, in this class I am going to start with fermentation, that is anaerobic respiration. Okay, what happens actually? Glycolysis occurs in any type of respiration, that is either aerobic respiration or anaerobic respiration. First glycolysis occurs, that means glucose undergoes partial breakdown into two molecules of pyruvic acid by a pathway called glycolysis that we studied in the previous video. Okay, so after formation of pyruvic acid, in the absence of oxygen, this pyruvic acid enters into fermentation, that is anaerobic respiration. If oxygen is available, it enters into aerobic respiration. Okay, now we are going to study about this anaerobic respiration, that is fermentation, which occurs in the absence of oxygen. Right. So before starting with this fermentation, let's recollect very few points of glycolysis. So what happens in glycolysis? Glycolysis consists of two phases, first and second phases. First phase is called as energy consuming phase and second phase is called as energy releasing phase or payoff phase. So what happens during energy consuming phase? Glucose will be converted into two molecules of 3-PGAL by the end of energy consuming phase, that is by the end of first 5 steps. So during this energy consuming phase, two molecules of ATP is consumed. Now, these two molecules of 3-PGAL which is formed, 3-phosphoglyceraldehyde which are formed by the end of first phase of glycolysis, energy consuming phase of glycolysis enters into second phase, that is energy releasing phase of glycolysis. So in this energy releasing phase, it releases energy in the form of NADH2 and ATP. So let's see where NADH2 is formed and where ATP is formed. So NADH2 is formed during the conversion of two molecules of 3-PGAL, 3-phosphoglyceraldehyde into two molecules of 1,3-bis-phosphoglyceric acid. What is this step? Sixth step, that is oxidative phosphorylation. So in the oxidative phosphorylation, what happens is 3-PGAL undergoes oxidation or breakdown. Due to this oxidation, breakdown or dehydrogenation, what happens to redox equivalents? That is protons and even electrons are released, which are transferred on to NAD+. So this NAD+ is reduced into NADH plus H+. And in this step, two water molecules are utilized and inorganic phosphates are utilized in the form of H3PO4. As inorganic phosphate is transferred onto this 3-PGAL, it is converted into 1,3-bis-phosphoglyceric acid. Previously, it was having phosphate group at third position. Now, due to the addition of one more phosphate is added at first position, first carbon atom of this bis-PGAL. Bis means by two phosphates, bis-phosphoglyceric acid. Now these two molecules of bis-phosphoglyceric acid undergoes through a series of reactions resulting in the formation of two molecules of pyruvic acid, that is 3-carbon compound, during which 4 molecules of ATP is produced and 2 molecules of water is released. Where this 4 molecules of ATP is produced, which is produced through SLP, substrate level phosphorylation, where at 7th step and 10th step. Okay, at 7th step 2 molecules of ATP is released and at 10th step 2 molecules of ATP is released. Whereas these 2 molecules of water is released during 9th step, that is dehydration. Right. Now after formation of 2 molecules of pyruvic acid by the end of glycolysis, what happens? In the absence of oxygen, these 2 molecules of pyruvic acid enters into fermentation, anaerobic respiration. And this fermentation is of two types, that is alcoholic fermentation and lactic acid fermentation. Now let's study this alcoholic fermentation and lactic acid fermentation. So this alcoholic fermentation occurs in yeast cells and some other organisms. What is this yeast? Unicellular fungi, unicellular fungus. Okay. So during this alcoholic fermentation, 2 molecules of pyruvic acid first undergoes decarboxylation into 2 molecules of acetaldehyde. Decarboxylation means removal of carbon dioxide. So this carbon dioxide is removed. As there are 2 molecules of carbon dioxide is released. And this decarboxylation is catalyzed by an enzyme, pyruvate decarboxylase. And this enzyme functions or it gets activated in the presence of TPP, that is thiamine pyrophosphate, and magnesium ions. So by the end of this decarboxylation, 2 molecules of acetaldehyde is formed. So when this carbon and 2 oxygens are released, that in the form of carbon dioxide. What will be remained? CH3, C double bond O, and this H will be remained, hydrogen. So this is the structure of acetaldehyde. Now these two molecules of acetaldehyde, now they undergo reduction into 2 molecules of ethyl alcohol. So during this reduction, what is reduction? Addition of protons and electrons. Protons and electrons, that is reduction. So these protons and electrons are donated by NADH. So 2 molecules of NADH undergo oxidation into NAD plus by donating protons and electrons to acetaldehyde. So that acetaldehyde is reduced into ethyl alcohol by receiving protons and electrons, whereas NADH plus H plus, by donating 2 protons and electrons, it has been oxidized into NAD plus. So NADH is oxidized into NAD plus by donating protons and electrons, whereas acetaldehyde has been reduced into ethyl alcohol by receiving protons and electrons. And the step reduction is catalyzed by the enzyme alcoholic dehydrogenase. Okay, now this is the structure of acetaldehyde. So how it will be converted into ethyl alcohol? So one bond, here one bond among these two double bonds, these two bonds, one bond undergoes breakdown, and 2 protons are added. Here one proton, here one proton. So that is the structure of ethyl alcohol. So this is about alcoholic fermentation. So the 2 molecules of NADH plus H plus, which are synthesized during glycolysis, which are released during or formed during glycolysis, are utilized during fermentation, that is alcoholic fermentation. So there is no net gain of NADH2 during anaerobic respiration. Okay, here I would like to discuss one more point here. So if the concentration of this ethyl alcohol reaches to about 13%, if the concentration of this ethyl alcohol reaches to about 13%, it results in the death of yeast cells. In NCERT, one point is given, one line is given. Yeast poison themselves when the concentration of alcohol, when the alcoholic concentration is reached to about 13%. Okay, so in case of alcoholic beverages like beer and wine, alcohol concentration will be lesser than 13%, and these beer and wine beverages like beer and wine can be produced by natural fermentation by yeast. Whereas some other alcoholic beverages like whiskey, brandy, rum, which are with higher concentration of alcohol, cannot be produced by natural fermentation alone. They are produced by distillation. Okay, so this is about alcoholic fermentation. Now let's start with lactic acid fermentation. This lactic acid fermentation occurs in lactic acid bacilli, bacteria called lactic acid bacilli, and even in human muscles in the absence of oxygen. Human muscles in the absence of oxygen. Okay, so during this fermentation, the 2 molecules of pyruvic acid, which is the end product of glycolysis, undergoes reduction directly without any decarboxylation, like as in case of alcoholic fermentation. So it undergoes reduction directly, resulting in the formation of 2 molecules of lactic acid. So this pyruvic acid, this is the structure of pyruvic acid. So during this reduction, what happens? During this reduction, 2 protons and electrons are transferred onto pyruvic acid so that it is reduced into lactic acid. Okay, so these 2 protons and electrons are donated by NADH in the same way. So by donating 2 protons and electrons, it has been oxidized. NADH has been oxidized into NAD plus, whereas by receiving 2 protons and electrons, pyruvic acid has been reduced into lactic acid. And the step of reduction is catalyzed by the enzyme lactate dehydrogenase, which is active or which is functional in the presence of FMN, flavin mononucleotide, and zinc. Right. So during this reduction, what happens here? This is the structure of lactic acid. So this double bond undergoes breakdown. So here there is no double bond. Double bond undergoes breakdown, and hydrogens, 2 hydrogens are added. Here one hydrogen and here one hydrogen. See here, here, and there is no double bond here. So this is the structure of lactic acid. So this is about lactic acid fermentation, which occurs in lab bacilli, that is lactic acid bacilli, and human bacills in the absence of oxygen. Okay, in the next video, this is all about fermentation. In the next video, I'll be explaining about output or balance sheet of this fermentation with respect to the release of ATP, carbon dioxide and water. Hope you understood this concept very well. Thank you, students.